like break this down a little?
Sure. So let's go over the 5 steps of. Actually, let's do this. I know it. See how well you know it before we go into it. 5 steps. Okay, I'm going to write them down. Put them in order. And then either one of 3 things. Write out a couple of sentences for each of those, what it is, or what's happening. Draw it if you want to, or talk it over with somebody else. We'll go 5 minutes. Order. What's one? Devin. Okay. You want to give me a description for attachment? Okay, so when the bacteriophage binds to the host cell, does anybody want to add anything to that definition or description? Erica?
It uses its tail fibers and base plates to attach to its.
Good. Tail fibers, I think, is a really important one. We'll come back to the base plates because the base plate has more of a role in the penetration step, but excellent. What is it attached to? What's that? Possibly. Some bacteria do. In the case of gram-negatives, there are some bacteriophages that actually attach to the O polysaccharides. What else? What was that, Claire?
LPS?
LPS would be part of the O polysaccharide. Others? Sorry, I didn't hear all of them. Maybe slime layer? Protein. Protein is the easy one. Outer membrane proteins, other integral membrane proteins. Possibly fimbriae or flagella, but in general, protein. Okay. I think that's good. That really helps out. Next up, penetration. Description? Kaylin? Penetrates. That's a great start. What penetrates?
Nucleic acid.
Yeah, just the nucleic acid. And at least in the case of bacteriophage, it's only the nucleic acid that's making it into the cell. That's something different from human viruses, but we didn't cover that. Don't worry about it. Okay. How does that DNA get from the capsid into the cell? Base plate does what? What was that, Matt? It lyses the cell wall, right? It's got lysins or something like a lysosome on the end there. Helps to hydrolyze that cell wall, creating then basically what's happening at that point is that there's this inner tube that kind of pushes its way through the membrane, or through the peptidoglycan. It's kind of spring-loaded, pushes its way in, creates a conduit for that DNA from the viral capsid head to move through into the cell. Okay, so that's step 2. 3. Biosynthesis. What's happening here?
Is it where it's taking the host DNA and the viral DNA kind of mingling?
Possibly. If we're talking about a lysogenic phage, we might see integration at this step. This is kind of at the point at which lytic replication and lysogenic replication split from each other. Another word for biosynthesis would maybe a term I would use would be central dogma. What's happening when we say central dogma? What events are we talking about? Transcription, translation, and DNA replication. Beautiful. We get viral genomes replicated. The viral genomes are being transcribed, and those transcribed products are being translated into protein. So that biosynthesis is producing the genetic material and producing the proteins that are required to make more viral particles. Okay. We'll come back to lysogeny. I want to make sure we hit on that too. Okay. So that's, oh gosh, that's biosynthesis. I'm going to break that thing soon. Not by choice, maybe. For assembly, sorry, jumped ahead here. What's happening during assembly?
Transduction.
Not a bad guess. But not in this case. So maybe finishing up protein synthesis. Big thing is, the example I brought up was that what's happening during assembly is basically, as the name would imply, all the parts of the virus have been produced. Now it's just a matter of bringing them all together. So the proteins and bacteriophage genomes, I should mention, bacteriophage probably encode on the order of about 50 to 100 different proteins. So, and the majority of them actually make up the capsid structure itself. So you've got these proteins that are required for the capsid head now coming and assembling together. You've got the tail fibers coming and binding with the base plate. You've got the sheet that makes up the kind of the body of the phage also coming into contact now. And all throughout this process, the DNA is somehow getting captured into the head of that capsid. So that's the assembly step. Just putting all the pieces together to produce the new variants or the new viral particles. Last step? The exit. And how does exit occur?
Cell lysis.
Yeah, exactly. Perfect. So the cells end up lysing, and one of the terms we used was lysins. Again, bacteriophage produce lysins that allow for that exit step to occur. Okay. So those are the 5 major steps. Be able to describe. I think that's really the main takeaway point from this, is be able to describe what's occurring during each of these steps. If you like the picture and if that helps you to kind of visualize it, great. But you do not need to memorize that picture. It's not necessarily a helpful activity. Okay? So before we go into talking, I guess, deeply about lysogeny, what are the differences between lytic replication and lysogenic replication? And I'll give you guys another 2 minutes. There's probably 2 or 3 things that are different between the two of them. So let's see what you can come up with before we talk about it. Started with this. Lytic versus lysogenic. What? Oh. Olivia. Good. So in lysogenic, the phage genome is integrated into the hosts. And then the other part that you brought up is it stays there. It stays a part of the bacterial genome until some sort of stress occurs. And we talked about that stress as being something that causes maybe damage to the DNA. And we didn't go into it much more depth than that. Okay? So another way to put this would be that stress reactivates the virus. And when we say that it reactivates, what do we mean? What's actually happening here? And just think of the process. So the viral genome pops out of the host genome, and what happens at that point?
It has to reattach.
Think at this point, actually, the viral genome is already inside the host cell. Okay? If you remember the picture we had, we talked about how lysogenic, so you get integration, it stays in the host genome. Every time the host genome replicates, the viral genome is replicated along with it. Eventually, stress hits, the viral genome comes out, and what happens next? Back to the lytic cycle, exactly. And it's not all the way back to the beginning. It actually enters right into the biosynthesis step. So it's not as if the infection is starting all over again. It's already gone through, the virus technically has already gone through the attachment and the entry. Now we're just jumping right into biosynthesis. So that's what we mean when we say reactivation. And then it would complete the assembly and the exit at that. What else? What are other features? Wait, sorry, go ahead, Matt, and then Caden.
When it's lysogenic, it can be passed on to this generation.
Okay, is that what you were going to say too? Okay, good. I'm going to write this down real quick. Another way we might put that, it's actually passed by vertical gene transfer, right? In all honesty. Go ahead, Caden. Yeah. And I'll put the infected cell survives. I mean, if folks are having a hard time understanding how this occurs, the best analogy I can give is if you have had, I bring up this example, but now I'm realizing probably only like 2 or 3 of you have had this. If you have had chicken pox, okay, or if you have had mono, or if you have had, those are probably the 2 most common. If you have had herpes virus, so cold sores, all of those actually, it's not lysogeny, we call it latency, but it is the same principle. Whereas inside our cells, the viral genome remains until some sort of stress occurs, and then you get a reactivation of the lytic life cycle for a brief period of time, and then those cells infect new ones and can go back into that dormancy phase. Okay? So there's some overlap with what we see in human viruses as well. Yeah, or latency is that lysogenic. It's basically like a dormancy. Exactly. Exactly. So like in the case of a cold sore, that's the physical manifestation of that lytic replication. Absolutely. Emma.
Is it true that you can only get chickenpox once? Is it because, or is that?
So, yes and no, right? So the answer is yes. Typically, we only get chickenpox once, and there's a couple of reasons behind this. One, our immune system is built to, anytime it sees the virus again, it's going to kill it before the full infection occurs. 2, the virus will stay in your body for the rest of the time. So at least for somebody like me who actually got chickenpox, and I haven't gotten the shingles vaccine ever, but oftentimes the second round of of the same viral infection from a reactivation event is shingles. It presents in adults as shingles instead of chickenpox. So where this concerns all of you, if you guys want to answer, if you don't, that's fine, but who's gotten the chickenpox vaccine? Okay. I bet I can age everybody based on this answer too, but the chickenpox vaccine, what we're finding, just real quick side note, folks who have gotten it are actually getting shingles at younger ages than what we've seen in the past. So you guys are screwed, in other words. No, but I mean, in all honesty, what they're saying is we think we need a booster shot now for it. Yeah, that's kind of where they're going with it. So. And shingles is apparently very painful. Yeah. Yeah, my dad had it in his 30s. Okay, sorry for the digression. Anything else on lysogeny or lytic life cycle? What's that? Well, I know we've kind of covered it in part, but I mean, is there anything we want to contrast here with lysogeny?
Does that like the lytic cycle occur more often?
Yeah, absolutely. So lysogeny is kind of a rare event, right? And one of the things that Caitlin and I were talking about, not all bacteriophage undergo a lysogenic life cycle, but every single one of them will undergo a lytic life cycle. What is another difference? Matt? Yeah, no integration into the genome. And so there's no free ride for the virus in the same way as there is with the lysogenic life cycle. Other differences? Olivia? Yeah, I mean, the way that I describe it is your lytic is a sprint. Lysogenic is more of a marathon. Playing the long game. How about the host cell? Dies in the case of lytic, yeah. Any others? Those are kind of the major differences, I think. Good job. Okay. More questions on this. I have an idea of where I want to go next, but I want to first see if there are any questions. Okay. We're going to transition into horizontal gene transfer because there are some similarities here with transduction. But before we do that, Whitney was asking, how much depth do you need to know for the different virus or for the different bacteria that we've covered? If you've seen the case study, the only one you actually really need to know about is what? Staphylococcus aureus. Okay, so the rest of them may be important in your daily lives, considered an enriching moment of the week, but for the exam, don't worry about it as much. Okay, so Whitney, does that help? And I'm going to double check the exam to make sure that that is correct.
You don't need to know about like Carini.
No, you don't need to know about Carini. I mean, honestly, if I were to list for you the ones that I think are most important, it's the lactobacilli, it's the staphylococci, and it's the enterobacteriaceae. But on the exam, staph is the conversation. Okay. Now, I need to make sure that I make a point clear, though. That does not mean that what we talked about on Monday, because we talked about other bacteria, but we also talked about other stuff too, right? We talked about opportunistic infections. I'm not asking you to know everything about C. diff, but what I am asking you to be able to do is to take the concepts that we talked about on Monday and, for example, say, what is the effect of antibiotics on the gut microbiome? You don't need to know anything specific about the bacteria to answer that question, though, okay? So I just want to make sure we're all on the same page here and people aren't kind of coming up during the exam going, you told me I didn't need to study. I said nothing of the sort. Okay. Matt. I mean, don't worry about it. It might be, let me put it this way. You might, for me, I think of it as you would probably be able to fully answer the question without knowing that information. But if you think it's going to help you in enhancing the answers, if you want to just have some of the pieces of that information as to help you out, go for it.
What was his question, Zach?
So Matt's question was, so you don't want us to know like the general characteristics of the bacteria that are in the gut, for example, right? And I'm going to rephrase that in saying, like we went through in class today, if I say the stomach's acidic, what kind of microorganisms are you going to find here? Yes, I do want you to tell me there are acetophiles, okay? But I'm not going to say, I'm not going to ask you what type of these 6 bacteria, what one will you find in the small intestine? I'm not going to do that, okay? So does that help, Matt? Okay. All right, shall we move on to horizontal gene transfer? Talk a little bit about that. All right. Let's start this one out the same way we started out the last conversation. 3 types. There's conjugation. There is transduction. And there is transformation. What I would like you to do, I'll give you guys another 5 minutes to kind of work on this one to start. Jot down some notes. Either describe what's happening or what are some important terms that are associated with these processes. Okay? 5 minutes. Go. All right, so back to the terms. So somebody walk me through this process. We've got 2 cells, a happy little couple. One is Mr. or Mrs. Whom? F plus. Okay. The other is Mr. or Mrs. F minus. What does the F plus cell have that the F minus doesn't? Pilus, or ability to produce a pilus. What else? The F plasmid. Anything else? Those are kind of the biggest features, okay? And then what happens? One of the 2 strands of the plasmid gets nicked, and what happens then? It moves, right? So it kind of unrolls. And that piece of DNA moves into the F minus cell, gets replicated, circularizes back together. Oop. And now both cells are F plus. Perfect. Okay. Any questions on that one? That's kind of the general theme. Let's see. I'm going to go back real quick to just see if there are any terms we missed that we probably should include. Oh, probably ORI-T. ORI-T would be where the nick occurs, the beginning of that transfer from one cell to another. What's that? The nick on the plasmid that's in the F positive cell. All right. That's, oh, Allie, what's up? That's with the HFR. So in the case of the HFR, I'll just cover this real quick for basic information so that, again, there's no confusion here. You have the bacterial genome, and then the F plasmid, or the remnants of that F plasmid, are integrated into the genome. Nick would actually occur at the chromosome, and then you start to transfer the chromosome from one cell to another. But oftentimes, you see where I've drawn where this nick occurs? It's kind of like right in the middle of that F plasmid. So imagine you'd have to have all 5 million base pairs plus the remainder of that F plasmid to be able to somehow transfer from one cell to the next. It takes a really long time to do that, and oftentimes that pilus breaks before that full transfer step can occur. And so what you'll find is then oftentimes these F minus cells that are receiving that DNA from an HFR will not get the genes that are required to, for example, create a pilus in the first place. So they still remain F minus. Does that help? Okay. Dana. We kind of mentioned like there's three parts to these F plasmids. One is the ORI-T, one is the gene to encode the pilus, the third is the endonuclease. A nuclease is basically any enzyme that cuts or destroys DNA. Basically, it hydrolyzes the sugar phosphate backbone of the DNA. So that is what creates the nick.
Okay.
It's just the endonuclease that's on that plasmid. Meg.
So for the exam, would we have to know like all the steps to each of these? Or is it just like the
I think it's the general concept. Let me think about this real quick, because at no point am I asking you to walk through step by step and describe it. So I think it's more the general concept. Okay, Grace.
So when the like plasmid is edited, is it already replicated or does it replication occur after?
Yeah, so I mean, you've got a double-stranded DNA to begin with, right? One of the two strands gets nicked, and it kind of gets unrolled. So then you're left with two single strands of DNA. Then you have to get replication to create the double strand again. So it occurs. It actually occurs once that transfer from one cell to the next has been completed. Other questions? Those are all good ones. Allie?
When are like all three of these processes like,
What do they all have in common?
no, like when do they happen? Like,
Oh,
just like randomly?
yes? No? I mean, what causes them to happen? What causes a man to fall in love with a woman or another man? The world cannot answer some of these ridiculous questions. Not saying that one is ridiculous. No, I honestly, these are events that happen all the time, but I couldn't tell you just by like looking at if I could look at bacteria on a plate right now, I would not be able to tell you, oh, yep, they're undergoing conjugation. I wouldn't be able to. It happens all the time, though, because the genes are there to allow it to happen. So in other words, if an organism is F minus, this will never happen. But what the environmental cues are that allow us all of a sudden begin to produce a pilus, actually, that's a really good question. I don't have an answer for that. In transduction, it requires a phage to infect a cell. And that is going to happen all the time. It's the same way that we get infected by viruses throughout the year. Bacterial cells will come into contact with bacteriophage, and if it dies, a transduction event could occur in those cases. In transformation, there are only some bacterial cells that are quote-unquote naturally competent. In other words, that can pick up DNA from the environment. And there are benefits to this. In some cases, it's kind of like you could consider it a smorgasbord. The ability of bringing in DNA into the cell, you chop it up into small pieces, and now you've got nucleotides, and you haven't expended any of the energy you need to synthesize those nucleotides in the first place. So in some part, these are kind of like ingrained into the cell as a process that occurs for the, I guess you could say maybe like for the life of the organism. But the really good one, I think the really good question out of that I have no clue about what the answer is what triggers the pilus to form? What triggers an F plus cell to all of a sudden begin the mating with an F minus cell? Maybe it's the pheromones from the F minus cell. I don't know. It's a good question, though. Does that help a little bit?
Yeah.
I'm sorry, it's not the most satisfying answer, but... No, not necessarily. Know that it happens. Yeah. All right. So that was, oh, Matt.
So you just said that it's beneficial for the cell to perform transformation because it saves its energy.
It does require ATP to bring them in. You're absolutely right. And it would cost ATP to break them down. But the amount of energy that it's expending to do that would be less than the energy it takes to make those nucleotides from scratch. All right, let's go through transduction. Or should we, let's go in the order that we had it. Let's go transformation then. So transformation, cell, finger drawings now. Cell has some sort of DNA transport protein, active process. Brings that piece of DNA from the environment into the cell. And here is the current genome of that bacterial cell. One of the things that we mentioned is there has to be homology. All right, so if there is homology between the bacterial cell and that piece of DNA that it's bringing in, we might get a recombination event to occur in which, and we have not gone over the specifics of this homologous recombination by any means, but basically it's like swapping one piece in for another. Okay, so usually the way that we draw that is just to kind of draw X's at where these homologous recombinations are going to occur. DNA from the current genome comes out, new piece of DNA goes in. Right. And in the end, and that piece of DNA that's come out at this point then gets broken down. Okay, it's going to get just chopped up into small pieces by nucleases, and those nucleotides can be reused for the next round of replication, for example. Devin. Say that again? Oh, are you thinking the blues that I drew on the end are getting chopped off? Those would actually get recombined as well. Those would get recombined as well. And usually what it requires, Devin, or what we find is that kind of point this out real quick. So what we find is that actually this is double-stranded DNA. I've drawn it as single-stranded, but those double strands actually separate from each other. And you get base pairing between this piece right here and one of those two single strands. So you actually get base pairing first. You get polymerase to come in and sit and finish the replication of this piece that is unique. And then this piece is already base paired as well. So you basically are kind of like, it's almost like filling a pothole in some ways. You're just kind of filling that extra space, and then it becomes integrated into the genome. So that would be the piece of DNA that has been removed or excised from the genome to make room for this new piece going in. Yeah. These are the questions I wish you guys would ask during class. This is so much fun. But that's okay. Beggars can't be choosers. All right, so that's 2. Transduction. Bacterial cell gets infected by phage. Those new phage they're super big ones. Most of the time, will incorporate their own genome back into their capsid structure, but one of the things that happens to this bacterial cell, in addition to being infected, that genome of it gets chopped into smaller pieces. And that is part of the viral life cycle, something we didn't necessarily cover. So in other words, the events that lead up to transduction are a lytic life cycle of a bacteriophage. Once that bacterial genome gets chopped up, if by some mistake, low frequency mistake, that one of those bacteriophage or particles takes up a part of the genome, now this is what we would call a transducing phage. Sydney. Honestly, the goal is just to chop up the host DNA. In other words, the purpose is that the virus, by doing that during its biosynthesis, ensures that the host can't transcribe its own genome. It can't replicate its own genome. So those enzymes or those proteins all get kind of like repurposed to making more virus. It's kind of like how Kathleen talked about, one of the things she mentioned was influenza shuts down host transcription. The benefit of that is that it basically encourages all of those transcriptional parts, or all of the, including the nucleotides, it repurposes them all to making more virus. Okay. Allie. Virus, it's in the viral genome, and so it has the virus usually to break apart the host genome like this. The virus has it in its own genome. And again, we're using the term nucleases very loosely here, but we see them a couple of times. So then the transduction occurs. So that cell bursts from lysis. Phage attaches, penetrates, new piece of DNA comes in. Again, if there is homology between that new piece of DNA and the host DNA, we might observe a recombination event, and now that gene has been moved from one bacterial cell to another. One key thing to remember about all of these, they can all lead to transfer of genes. Not only just new forms of the same gene or different alleles, but also completely new genes into the bacterial genome. Ones that are not there in that bacterial, in the bacterium's ancestors. Okay? And that can lead to the spread of antibiotic resistance. Meg. Okay, yes. So yes, it can lead then to antibiotic resistance. Some of those genes might be resistance markers. Questions?
You're saying the bacterial genome with that needle strain put in the first virus, that happens all the time?
Let's put it this way. I can see it in the lab happening about once out of every 10 to the 9th or 10 to the 10th bacteriophage. So it happens. It's rare. In most infections, in most lytic infections, actually the vast majority of them, this will not happen. It's a mistake.
But you're saying that it's part of transplant. Like 100% of the time it's
Yeah.
So then does that one virus lead to the cell that's infected with the bacterial genome?
Yeah, and the rest of the viruses would leave that cell as well, but they will perform the normal round of viral life cycle that we would expect. This is just kind of a rare event.
So that's why you have in the first cell the bacterial genome as well as the recombinant.
Exactly. Okay. Can we take a quick 2-minute break, stretch, and then, or whatever you want to do, go to the restroom. Then when we come back, let's work on DNA, RNA for a little while. Nucleic acids. Few things that you need to keep in mind. The first are the properties of antiparallel, complementary. What do we mean by that again? Anybody recall what those two terms mean? If nobody answers, I'll sit here and eat peanut butter and dance. Devin. Good, that's the antiparallel. So if this is reading 5 to 3 here, that means the other strand would be in the 3 to 5 direction, left to right. Exactly. What else? What's complementary mean? That's our base pairing. Perfect. AT or AU, GC. Perfect. Okay. That top strand there, what kind of nucleic acid is it? 2 flavors. Not template or not template. Sorry. DNA or RNA? How do we know it's RNA? It's got the uracil. Perfect. Okay. I think those are the basics that we need to cover before we move on to the next step, which is how do we correlate these with each other? Now, always remember that when it comes down to mRNA, the mRNA when we are going from, when we are trying to read a codon, a 3-nucleotide sequence that encodes for amino acid, we must be reading it in the 5' to 3' direction. It must be the mRNA that you're reading. Okay? So in other words, the way that I have written at least 2 of these is not the RNA. And the question becomes, well, what the heck is the relationship? In other words, you've got to convert these to RNA. Okay? So what I'm going to do, well, how about we start with this? What is the relationship between RNA and tRNA? And use the terms that we've talked about already. Yes.
tRNA is something that brings the amino acid sequence.
tRNA is the thing that brings the amino acids to the ribosome. We oftentimes say the tRNA base pairs with the mRNA codon. In other words, these are the anticodon. Does that help anybody now to realize what the mRNA sequence might be? Or what the relationship is? What would the mRNA sequence be?
The codons?
Yeah, so the codon that binds to this. I heard it, GCC. What end is over here? 5' GCC. How about this one? 5' GGC. In other words, the relationship between your tRNA and your mRNA, the tRNA anticodon and the mRNA codon, terms we've already used. They're antiparallel, they're complementary. Okay? Antiparallel and complementary. So go ahead and let's write down that one real quick. The arrow here is just meaning that there's a nucleotide change. That's what I'm trying, and we're going to actually look at how does this change the amino acid sequence. Sydney.
I missed this when you said it, but how can you tell between the non-template and the template?
You can't unless I tell you. Yeah. So the above one that I labeled as non-T, that would be a non-template strand. I would tell you that. Yeah, you can't tell that, at least given what we've talked about, there's no way I would expect you to just know that on your own. I would tell you ahead of time. So like this bottom one, I'm telling you it's tRNA. Where your mind should go is, okay, the mRNA is complementary to that tRNA. It's antiparallel. In other words, and these arrows are meaning there's a mutation. That's the other part of this that we'll cover.
And that's the mRNA.
So this here in red would be the mRNA sequence. Exactly. Okay? Devin and then Allie.
So, to like, just like to clarify, when something is like complementary, then it'll be complementary to the template strand?
It could be it's complementary to whatever the other strand is. So you have to put you always have to know what the context is. So for example, the mRNA is complementary to the tRNA sequence. The mRNA is also complementary to template DNA. Okay? We'll map this out in a minute. Maybe it will help to actually kind of like draw some arrows and show what this all means. Maybe, should we go to that first?
Can you clarify when you're saying non-template and template? Like what change the meaning?
Absolutely. So let's map this out. Okay? 4 major parts. tRNA, mRNA, template DNA, non-template DNA. Now, template DNA is the strand that gets transcribed. In other words, as the RNA polymerase reads the template DNA, it is producing mRNA. Okay? I'll draw a quick picture. So template DNA. So let's just say those are 6 nucleotides in the template DNA. Okay? RNA polymerase is coming along. What is the mRNA sequence going to look like? Okay, U, G, A, U, C. What am I missing? Thank you. I'm missing the 5' and 3'. What direction does what does an RNA polymerase make the new DNA in? Oh, yeah. It makes the new RNA in the 5 to 3. Okay, so that polymerase is always moving in the 5 to 3 direction of the new strand that's being made. In other words, what direction is it moving on the template? 3 to 5. Good. Okay.
And that's, you said it made mRNA?
Yep. So this top strand here would be our mRNA. In other words, let's go back to this. What we've just modeled is the relationship between template and template DNA and RNA. I need abbreviations, but they're anti-parallel, AP, and they're complementary. That is the relationship between them. Okay. Any questions? Oh, go ahead, sorry.
Sorry, what are they?
Anti-parallel. Allie.
The circle, is that the RNA polymerase?
Yep.
Okay.
Shuffle. Complementary, anti-parallel. Sorry, I'm just getting tired of writing words right now. I'm lazy. All right. What is the relationship between the tRNA and the mRNA? They're anti-parallel and complementary. We just established that with the past problem, right? The tRNA has the anticodon, the mRNA has the codon, so that is another one. That is anti-parallel and complementary. Huh? Yeah, I can actually. Can you? Sorry, that was passage. That's what I was. So the tRNA.
RNA is made from the mRNA.
So of course, this would be in the this is gone now. This would be in the context of a ribosome. But that tRNA anticodon would be, here's the 3' end of it, U, C, A from 5 to 3, and off of that 3' end, that's where our amino acid is located. And that's not something we covered, but that's how the amino acid actually gets brought to the ribosome. It's physically attached to the tRNA at this point. Okay. Does that help?
Yeah, so you just match up the codon.
You just match up the codon with, yeah. And again, anti-parallel and complementary. Okay.
So the mRNA is going from 5 to 3, so the tRNA is always going to go from 3 to 5.
Yeah, if we were to read it this way, yes, yes. Now I say that because I could be a complete asshole and actually change the mRNA sequence. Oh damn. Well, guys, it happens all the time. Just saying. Okay. What is the relationship between the tRNA and the template DNA then?
Well, the tRNA just goes out. Is it basically the same, just by changing the units?
Perfect. It is the same.
What was your statement?
The question was, what is the relationship between the tRNA and the template DNA? In other words, and what Meg correctly said is they are the same with the exception of T's and U's. That U's in RNA, T's in the DNA. So if we look at this, actually, I mean, this anticodon here, 3 to 5 direction, reads ACU. Well, the DNA, template DNA in the 3 to 5 direction reads ACT. Same thing, just T's and U's are switched.
So you could ask the question saying like the tRNA, you're given the tRNA not translated to template DNA.
I could. I could do that. All right. What's the relationship between template DNA and non-template DNA?
If you were to do that question, it would just be the same, we just swap out the T's and U's.
Yes.
Okay.
Yes. Yes. What is the relationship between template DNA and non-template? Devin?
They're complementary.
Perfect. They're complementary and they're anti-parallel. How about this one? mRNA and non-template DNA. They're not. They're the same. They're the same. Because think about it. Look back to this example. If this is our template DNA again, non-template DNA is the strand that doesn't get transcribed. It's just another strand there. So our non-template in this case would be 5' T G A T C.
So this is also just changing out the T's and U's?
Exactly. Exactly. So the relationship between the non-template and the mRNA, same.
So what's the point of the mRNA if we look at the tRNA to look at the next result?
You don't look, well, so that's one thing that I want to make sure we get straight here. The codon code is not based off the tRNA. It's based off the mRNA sequence. Okay? The tRNA carries the amino acid, but when we actually read that codon code, what we're reading is the mRNA. Okay? So, in other words, UGA, that's not a good example.
So like if you would give us something like that on a test, we would have to look at what UGA would be in the codon term?
Yep, exactly. Which is a stop codon, which is why it's a sucky example. But yeah, exactly.
When you're saying that's the same, you're saying that's the 5 to 3 prime.
Yep, and what I'm saying is both of them are in the 5 to 3 prime. The G's, A's, and C's are the same, but T's and U's we have to swap. Yep. So, if you're kind of thinking of a mental list of things to check for in this. 1, you could memorize what's anti-parallel and complementary. That's one way to go about this. But, I mean, we've just kind of mapped out 5 relationships right here. Okay? I would look at that and be like, that's a lot to memorize. One of the things, you might just want to start with the base. You might want to start with template DNA is used to make the mRNA. And if you can start there, then it's pretty easy to remember the rest of the connections. mRNA base pairs to tRNA. When we say that it base pairs, we mean that implies that the two of them are anti-parallel and complementary to one another. Template DNA. Base pairs to non-template DNA. Again, that implies complementarity and implies anti-parallel nature between the two of them. Okay? Sydney.
But RNA polymerase reads the DNA from 3 to 5, but it makes it from 5 to 3.
Exactly. It makes the mRNA in the 5 to 3. The other way to think about this is basically everything is opposite. If you can keep that in mind, that everything is opposite, then it might help, I don't know. Carla?
Okay, so the order would be not to put template mRNA TR.
If you were thinking about it yeah, that's one way to think about it. Yeah, that works really well, and each of those is anti-parallel and complementary to the other.
Figure out which way to go.
Yeah, exactly. Zach and then Meg. Okay, Meg.
So on a test, if you would give us the tRNA strand, we would have to put it into the mRNA strand in order to get the photo.
Exactly. So let's do that. Shall we try a couple now and take a look at it? All right. So here's a good sample equation or question. I'm going to get rid of this one real quick. I'm nice with the first one. It's mRNA. Second one is non-template DNA, and the third is tRNA. What I want you to do is, given these sequences, write the mRNA codon. And then if you have the sheets with you, like the codon code, what I'd like you to do is identify, and each of these are representing a point mutation. Identify the amino acid before the point mutation. Identify the amino acid after the point mutation. And then define what kind of mutation has occurred. Missense, silent, nonsense. Let's start there. And I'll give you like 10 minutes. Take the time to work through this. Okay? So I'll give you about 10 minutes to work on this one. If this ends up going longer than 6, so be it. Okay? I'm not going to rush out of here. So if we need some time to work on it, let's take the time to work on it and get it right. I'd rather put in the energy now than grade all of these with red ink on Monday afternoon. Okay? So, go ahead. AUU encodes for? What's that? Isoleucine. I-L-E. And AUG is methionine. I only know that because it's also the start codon. Devin. For the second one. So for this one? You're absolutely right, but what's the relationship? So that let me go through this real quick. So if we were to just make the jump from non-template to mRNA, it's the same sequence, okay? But the way that we would another way to think about this would kind of be to do the following route. Non-template DNA is complementary and anti-parallel to template DNA. Template DNA is what gets used to make the mRNA, and that relationship is anti-parallel and complementary as well. So I'm not going to say that what you're saying is incorrect. It's just it sounds like what you've done is you've written it or you've thought about it as the template DNA and not done the final step to the mRNA. Does that help? Okay. Another missense mutation. Okay. And then our last one here, tRNA starts 3' CGG. What is our mRNA? What are we missing? Which side? Okay. Amino acids for these two? What was that? ALA, alanine. And what's the other one? GLY, glycine. Okay, so all three are missense. Follow-up question. Which of these might be most detrimental, or which of these might be least detrimental to the cell? They're all missense. They'll change the amino acid sequence, but which of the three might be the least bad? Meg? Why?
Because their chains are similar.
Their chains are similar. And by that, what do you mean? So go a little bit more in depth for me. What do you mean by similar? And you don't have to say like the specific sequence, but what are some properties about them? They're both nonpolar. Perfect. So they're both nonpolar. The length of their chains is somewhat similar to one another. You don't have to count up all of them. But those properties of both being nonpolar, because it says so on the chart that I gave you. So Sydney, there will be an essay a question like this on the exam. I want to fairly point that out again. Okay? But on that sheet that I gave you, it actually lists out all of the amino acids and shows you the properties of them. And so you might want to take a look at that ahead of time just to be familiar with it. You don't need to memorize any of them because I'll give it to you again. But we want, if there were a missense mutation to occur, it's better to have similar amino acids in there than ones that are different from each other. Okay? Examples might be like, well, threonine and asparagine is actually not a bad example. How is threonine grouped? It's polar. What makes it polar? What's that? Well, all of them do. That's a common feature of all the amino acids. Sorry, so focus just on the side chain. What on the side chain makes it polar? It's got a hydroxyl group, and we know that the oxygen is fairly electronegative, right? What about asparagine? What's true about its side chain? It's polar as well, but what's different about it? What's that? There's not a hydroxyl group. There's an amino group. Anybody see a plus by it? At the bottom there? Is there one on there? Oh, shit.
Well, you have the oxygen, which is all it has. Is more.
Well, hell, that does. Okay, I guess I'm not going to ask that question then. No, not that specific one. I'm still going to ask the question. It doesn't. You're right. I thought it did. I was wrong. I was thinking arginine. Okay, never mind. That one wouldn't be that bad either then, apparently. Ones where you might find two polar ones being a bad thing, tyrosine to aspartate. Tyrosine's got an OH group. It's a polar. Aspartate at neutral pH, charged. Has a full negative charge on it. That does actually change the properties of it. Okay, so keep the concept of polar, nonpolar in mind. I'd expect you to be able to do that because of biochemistry, but yes, absolutely, absolutely. Given that we haven't covered that this semester, I don't know, I'd have a feeling that not everybody would get that one. But yeah, definitely you would look at that as a possible effect as well, because we've talked about it. I mean, anything with cysteine switching to something else, you lose the disulfide bond interaction that usually occurs between cysteines. Yeah, absolutely. Okay, I'm going to pause here. How are we feeling?
I'm feeling like that Monday option is getting.
God bless. Okay. I'm looking at time. I know Meg wanted to go over some antibiotic resistance stuff, just in terms of just general practices of antibiotic resistance and some of the principles here. So I do want to make sure that we have some time to talk about that. What I will be willing to do on my end, if you would like, is I can give another practice problem like this. Would that be fair?
Yeah.
To give one more practice. When we were talking about antibiotic resistance, some of the things we talked about were the practices. We also talked about practices that lead to antibiotic resistance. We talked about ways to get around or alleviate those practices as well. We also discussed, Maren McKinnon brings up some ideas of like big data tracking that we can do as well. So let's just start with the basics. what leads to antibiotic resistance? From a social perspective, not from the bacterial cell, but from a social perspective. What practices lead to antibiotic resistance? What's that?
Farming and agriculture.
Farming and agriculture. I heard ear culture, and I'm like, what are you talking about? Yes, farming and agriculture. In what respect?
The antibiotics that are
And oftentimes those antibiotics are being overused, or maybe at another thing we might bring up is they're being used at concentrations below their optimal. There's just a new study, actually there's just a new report out on this. World Health Organization is asking farms to stop using antibiotics on livestock. It just came out yesterday. Other examples. That's what they're asking. Probably, I guess, as a prophylactically. What else? Good. So using antibiotics when they are not useful. So using it for a cold rather than, which it will not work against, or using it for an infection maybe that the body can clear naturally. So ones that particularly that prescribers are at least the percentages seem to be reduced in the times that they are prescribing it would be ear infections, sinus infections are particular ones that antibiotics are being prescribed for less frequently now. Other examples. Not taking them for the full amount of time. So if it's a 10-day prescription, take it for 10 days, right? Or if it's expired. Exactly. And why was that one a problem?
Like some of the ingredients in the antibiotic won't be working, so they'll just gain
Good.
resistance.
Exactly. So in other words, the antibiotic, there's an expiration date on these because at least they've been tested to be stable until at least that date. And beyond that date, the pharmaceutical company can't guarantee that it is still, if it says it's 100 milligrams of an antibiotic, it can't guarantee anymore that it's still 100 milligrams after that time. Some of it may have been degraded or inactivated, in which case it might be 80 milligrams of active antibiotic. In other words, you're treating with a sub-inhibitory concentration, one that is less than required, that would be suggested as a therapeutic dose. Okay. Other examples for Emma.
Using broad spectrum.
Yeah, using broad versus narrow, and we said one of the issues with that, not only in terms of the microbiome, but wiping it out, but also, I don't know if we hit this one home really hard, but the idea that if you are using a broad spectrum antibiotic, any of the organisms that are being exposed to that broad spectrum antibiotic could gain resistance to it. Ones that might be pathogenic and ones that actually we could casually be turning into pathogens in some ways as well. Absolutely. Others? I think those are the major ones. I think that's a really good list of reasons. I do want to just kind of point out a couple of things again. So ideas that Marin McKenna has for reducing the frequency or controlling antibiotic resistance. Let's track. Let's track where the outbreaks, where these infections are occurring, or where these. So the 16S ribosomal RNA is thought to be like a molecular clock. So we use it there are a couple of ways in which it's used in microbiology. One is to determine how closely related are two species from one another. That's not the context of how we talked about it with relation to this paper that we looked at today. Okay? The way that we talked about it was we can actually use sequencing to identify a microorganism. And the regions that we want to sequence are those variable regions, because those are the parts of the 16S that are going to change the fastest over evolutionary history. Okay. And so by determining the exact sequence of an organism's 16S ribosomal DNA, we can get pretty close to identifying what species it is. We at least can determine in most cases the genus. We may not be able to distinguish between two species within the same genus. So in terms of like microbiome studies right now, if you think of the sheer magnitude of microorganisms, we said 10 to the 11th per gram of feces, for example. Nobody wants to try and identify 10 to the 11th microorganisms by biochemical testing. It is expensive. It is ridiculous to do. So what researchers will do now is they will just isolate the DNA, throw it into an instrument that actually just analyzes the sequence for them, and what they get at the back end are these printouts of, given this particular sequence, this organism is in your sample. Okay, so like if the 16S is identical to a known sequence for Staph aureus, it will say, you have a Staphylococci in your sample. And this is all, we're able to do all of this because we've done a lot of sequencing of organisms already, so we know what the 16S should look like. We're just comparing what do we get from a sample to what the known 16S sequences are for different bacterial genera. Okay. Does that help? No? Damn it. Erica? You know how we were looking at the microbiome stuff today? That's the context. So in other words, on these global scale studies where we are trying to understand what microbiome makes Erica, we sequence the bacterial DNA in order to figure out what bacteria is there. That's the purpose. And we've got catalogs of this information now. Now the question becomes, what do you do with it? And that's kind of the point where we're at with this research, is like a lot of people know what's there, but the question is like, okay, what the hell does it all mean? Nobody knows, really. And that's where, one of the things I was trying to point out today with the study is, that's where we're going. That's where research is going, is now that we know what's there, now that we know we can inoculate a baby, okay, and now we can figure out, does that inoculation actually have an effect? Do the particular microbes there have an effect? So if you are a child with a poor immune system, maybe what it would allow us to do down the road is we need to feed this baby probiotics. We need to feed it lactobacillus because we've learned through history that lactobacillus leads to the development of a good immune system, which would probably be much more socially acceptable than saying we need to take this kid and we need to shower him in vaginal secretions, right? I mean, much more socially acceptable. So that's where I think we're going with it, to kind of give you a bit more of a context. Yeah.
When you're talking about like the Staph aureus, obviously.
Mm-hmm.
So you said like that's the main bacteria that we use.
Yes.
And are you talking about like the grams and stuff like that?
Gram status, morphology, and then the couple of the other notes that I put. It's halotolerant. I can't remember what the other one is, but those are the basic features that it's like 4 bullet points of information total.
We need to know that for the studies?
You should know it, yeah. Instead of memorizing every single one of those microbes that I asked you to remember, just know the information about Staph aureus. That is the one where I want you to actually know some specifics, or where knowing those specifics might be helpful in the context of the exam. Grace.
Can we go over like the describing the effects of HPV?
Say that again?
Describing the effects of HPV and influenza and HIV on this.
No, because it's not going to be on the exam. Yes, it's not going to be on the exam. and I don't want to go over it.
Is that, like influenza and like the green and blue?
So yeah, that, and you should know it has an RNA genome. Those are kind of the most important features. Look, I think what she came in and did was great. I don't fault her for anything that she talked about. I think it was a really good conversation. Just some of this stuff, like when I look at it in retrospect, I'm like, is that the most important thing we need to be worrying about right now? And the answer is probably no. In all honesty, I don't think I need you guys to understand every single concept of influenza. There's a lot of cool stuff about it, but given the amount of time that we spent on it, it's a snapshot. It's not going to be that important. Emma.
What do you want us to know about antivirals?
Why they're challenging to make, I suppose. So, I mean, one of the things that Kathleen talked about with antivirals was the concept that what makes antibiotics so good is it targets things that are unique to the bacterial cells. What's true about the life cycle of any virus, any human virus? It has to go through attachment, penetration, yada, yada, but where is it doing its assembly? Where is it doing its biosynthesis? It's doing it in our cells. And so antivirals are challenging for a couple of reasons. One, the viruses infect our cells. So a true antiviral has to target something that is unique to the virus and not to us. The thing that's tough with viruses, at least human viruses though, most viruses have like 10, 20 genes total. So you're talking about a very few number of proteins that you could create drugs against. So that's a second challenge. Third one, and this is one that she brought up a little bit. Honestly, if we had more time, I'd love to spend time talking about this, but the mutation frequency. So viruses, RNA and RNA polymerases, they don't do as good of a job at transcribing as DNA does in replicating. DNA polymerase makes a mistake maybe once every 10 to the 6 base pairs. It has proofreading ability. RNA polymerase doesn't proofread. And so because of that, the RNA viruses mutate at a quicker rate, which makes some of these antivirals pretty ineffective after even a couple of years. And if you've ever been put on like Tamiflu, which is used for influenza, some people take it prophylactically. It doesn't work against all strains of influenza anymore because of the mutation rate. Yeah. Yeah. And as soon as it starts to mutate, it's just like an antibiotic. If it mutates to a point in a way that the antiviral won't bind to it anymore, then the antiviral is basically ineffective. That's why we get a new flu shot every year. And that may be a thing of the past as well. Soon. Very, very soon. Because I think they think they might have a universal flu vaccine. Yeah, pretty new. All right, well, I guess nobody wants to hang around anymore. Have a good evening. Devin. Yes, you may. So if you guys want to hang around, you're more than welcome to. I'll hang around for a few more. Otherwise, feel free to head out. Give us like a minute.
I just want to know when your office hours are next year. Not even for me, I'll just ask.
I will be around from 12 to 1 tomorrow, definitely. And I want to say 3 to 4, but I got to double check that because there's a good possibility I might have a meeting at that time. So let me just check that real quick. Nope, 3 to 4:30 would work as well tomorrow as well. And then for Friday, 9 to 10, and actually Friday lab. I will be taking John's place Friday in lab, so I'll be around during lab time too. But definitely 9 to 10 on Friday, and probably like 12 to 1, I think.
Thank you.
Thank you, guys.